Okay, hi guys. So today I will continue with chapter 21 biotechnology. Okay, so after 21, 22 would be the environment, like pollution, all those stuff. Then actually the whole chapter is done. And as I said, it's more on the revision of the earlier chapters. Okay, so I hope that you did your revision lah. And I'll quiz you endlessly next week onwards. Okay, so let me share screen. All right. Make sure you get this in your notes. I will check your notes when school starts. Okay, chapter 21, biotechnology. Okay, what is biotechnology? It's actually, you see, there's a word bio, right? It means life. Technology means, modern technologies, things like that. So using living organisms to carry out process that make substance that we want. So actually, we've been doing this for many, many years since ancient time. Just that maybe people from the past, they're not sure they are using a live organisms. Okay, for example, we use yeast to make yogurt and cheese, things like that. Okay, and then we also look through genetic engineering later. It actually means take a gene from one organism and putting into a different one. Okay, so this genetic engineering, there might be issues arise where people would fight against we shouldn't artificially change things. God made it like this way, things like that. For that part, I want you to read it yourself. Okay. Okay, so let's go through this one. The first one is DNA. Some of you are taught already, but just to revise back. Okay, DNA stands for deoxyribonucleic acid. I hope you all should know by now. Okay, and it's a chemical that makes up our genes and chromosomes. You know, right? DNA. So it's made of two long strands. You see that? Two long strands, and each strand there is a base there. Okay, so there are 4 base types. So there's A, there's C, there's G, there's T. Basically, there's like a code lah. Okay, now you need to know that T and A is always together, G and C is always together. You see, C, G, A, T. Now how to memorize this? I did tell my students to memorize the curve. So G and C are both curved, so they're stuck together. Okay, that's it. So you need to remember T and A is together, G and C is together. That's it for DNA. So sequence of base, so how the base aligns together determines what kind of protein your body will make. All right, now let's go back to using microorganisms. So biotechnology, genetic engineering often make use of microorganisms such as bacteria and fungi. Now, this is an important aspect, which is sometimes the essay question will ask you, is it okay to use microorganism? Why do we use microorganisms instead of using animals? Okay, so here are some of the reasons why we use microorganisms. For one, because the size is so small, okay, that they're easy to grow in the laboratory. You can grow millions of them in just one fermenter. Okay, it does not take up space, they reproduce very quickly, and you can make a lot of different type of chemical substance. And no one minds what is done to bacteria and fungi. Okay, if you come across a laboratory where they breed the microorganisms and they just throw the microorganisms away, things like that, you won't make a big hoo-ha, right? Because we don't really think like they are living organisms, but somehow we just don't mind. Okay, unlike if let's say animals, like a lot of people have issues with the animal farming, the ethical issues like slaughtering animals, killing animals, things like that. Okay, which presents more problem like if we use animals in scientific engineering for the benefit of humans. Okay, but no one minds what is done to bacteria and fungi. Unless you do, you can let me know. Okay. Third, we also share same kind of genetic material bacteria. As hard as it is to believe, okay, bacteria look so different than us, but then we do have some kind of same genetic material. So we can take a gene from human cell, we can put it into bacteria cell, no problem. Okay, and also bacteria have loops of DNA called plasmids. We will see this later. Okay, this plasmid is very easy. It makes things very easy to transfer genes from one cell to another, right? That's why we use microorganisms. Okay, now these are some of the examples of biotechnology. Okay, using yeast. Okay, you know that yeast is used in bread, cheese, or also making alcohol. Okay, yeast is single-celled fungus. Recap is a type of fungus. Okay, able to respire anaerobically. Recap, anaerobically means no need oxygen, right? Okay, just feed them glucose, and then they will start to break down glucose into ethanol and also carbon dioxide. Now, ethanol in your chemistry, you should know it's a type of alcohol. So this process is called fermentation, depending on what you want to use the yeast for. Okay, if you want to use yeast to make bread, then the CO2 part is very important. Okay, so when you put the yeast inside the dough, and you know, when the yeast release carbon dioxide, they are trapped inside the dough, isn't it? So once they're trapped inside, the dough will rise. You see why bread from something flat becomes fluffy and big? It's because of the carbon dioxide released by yeast. Now, some people will ask me, okay, since yeast produce ethanol, isn't the bread alcoholic? Actually, the alcohol is broken down because the temperature is quite high. When you break the bread, alcohol just is killed off. Unless you are making wine, you are making fermented things, then you just, you know, put the yeast together with the grapes and just leave them one side in the cooling place. Then yeah, slowly the grapes sugar will be broken down into wine. Okay, but no high heat involved lah. Okay, the next thing is enzymes, using enzymes like a biological washing powder. Okay, so the washing powder that you use, they contain enzyme and detergent. Okay, what is detergent? Basically, always use the word detergent, but detergent actually means something, a substance that helps the oily things, the grease, to mix with water, so you can easily wash things away. Okay, and the enzyme is the part where it helps to break down substance which can stain clothes, like oil stain, blood stain, things like that. Okay, right. So two type of enzyme. Okay, when you go through the enzyme chapter, I hope you already know what I'm talking about. So protease enzyme, remember, it breaks down protein. Okay, so having this inside washing powder is good because let's say you just killed someone, your clothes full of blood, you want to wash it away, no problem, use biological washing powder because blood contains hemoglobin. Hemoglobin is not protein, right? Okay, so proteins can break down hemoglobin, and the blood, you'll see that the blood disappears from the clothes because, you know, the molecules get smaller. Lipase is the one that breaks down fats, so it is especially useful if you're, let's say, cooking and all the oil grease just sticks onto your clothes. Okay, so the lipase enzyme breaks down the fat into fatty acid and glycerol. Okay, the main point is to make it very small and soluble so that it just goes throughout together with the water and your clothes will be clean. Okay, another enzyme is pectinase. Okay, well, how come commercial juices like Tropicana, okay, let's not use Tropicana, Tropicana or Sunkist, how come their juice is different than the juice that you make at home? Why would the juice that you make at home is still very cloudy, it still has fruit pulps? It's because commercially made juices in factories, they do use pectinase. Okay, this is an enzyme. Pectinase is an enzyme. Now it breaks down something called pectin. Okay, so fruit actually has substance called pectin, which help to stick plant cells together. So if you break down this pectin, then it's easier to get clear, nice juice out. Yeah. Okay, so this one is very important for paper 6. It always comes on paper 6. Okay, so I want you to know pectinase breaks down the fruit plant cell, breaks down pectin. Okay, right. The next part would be learning about penicillin. Now, a bit of history, just a bit of background story. I'm not sure what. I think it's diarrhea. So during the last time ages, there was this diarrhea outbreak, and people started dying from diarrhea, and because they lose a lot of water, they lose a lot of mineral salts, right? So there's a scientist who came across a cure when he fermented orange skin or something like that. We can research. Basically, there's this bacteria inside the orange peel called penicillium. Somehow, this penicillium, okay, the fungus penicillium can kill the bacteria of the diarrhea bacteria. Okay, E. coli, yes. Okay, and then after this discovery, right, he started to grow penicillium. So penicillium actually makes a substance called penicillin, and this penicillin is an antibiotic. And this antibiotic helps to kill bacteria and it doesn't harm human cells. Okay? So let's learn through the process of how we grow penicillium in a fermenter. Okay, now we need to look at this diagram. Okay, so this we call a fermenter. Okay? Basically, it's a very big tank where you control all of the environment for the fungus to grow in. Okay, and then you see this word called culture. Culture basically just means the liquid that you grow them in. Like Vitagen is actually, if you notice your Vitagen or if you drink Yakult, you will see that it says a culture drink. Basically, you're drinking in all the bacteria, good bacteria that are living in that liquid, that culture. Okay, so this culture contains carbohydrate and amino acid. Obviously, is to give nutrients for the penicillin. So let's just go through one by one. Okay, so this is where penicillium is added. There's this hole here and the sugar. Okay, here you have your culture. Okay, so your nutrients inside your culture, which includes carbohydrates, includes amino acids. Okay, now here it did not label, but this thing is actually called a water jacket. Okay, water jacket. It actually helps to cool down the temperature. We don't want the temperature to be too cold. We don't want it to be too hot either. Okay. Okay, and then when you look at this, there's some airways here, right? Okay, let's start here. So they do put steam in or sometimes cold water in, okay, because there is a thermometer inside actually. It's not labeled here, but there's a thermometer dictating like how hot, what is the temperature inside. We want the optimum temperature for the enzymes to work fast, to produce, okay, at optimum rate, right? So there is a thermometer there. So it's either if it's too cold, then they'll add in steam. If it's too hot, then they'll add cold water, okay, to control the temperature. Now, when the essay question asks you why is this so, you can say to maintain optimum temperature for highest rate of reaction, to produce high yield of products. Wow, 3 marks already. Okay, so this thing goes out. Okay, steam or water, cold water goes out. And here we have the air supply where there is a continuous supply of oxygen, okay, for the fungus, for the penicillin, okay. So you also, there's actually a stirrer inside. I don't know why this picture from your textbook doesn't show you, but in the exam paper, it will show you a stirrer, like a big spoon like that, okay. Now we stir it continuously so that the nutrients and the oxygen can be grounded balancedly, so all the penicillium is in excess of nutrients about oxygen. They don't fight for it, okay. Now, and after that, it usually takes 1 week because they produce so fast and they make all the things fast. You see all the penicillium, the penicillin produced also is inside the culture. Okay, so slowly they will release out the culture and then they collect the penicillin. Okay, important, Alisa, please remember all the parts. Okay, we're done with that already. I would like to repeat again and again. This is a very important question for paper 4. Okay. Okay, now we go to a genetic engineering process. Okay. Just now I did tell you that it is easy to transfer our gene into the bacteria. Now, why is this so important? Is because, you know, if let's say you want antibiotics, we want the gene, we want insulin, let's say insulin for diabetes patient, and this diabetes patient doesn't produce insulin, remember? So where does this insulin come from? Okay, in the past, we take insulin from live animals. Maybe we take from the horse, then we inject it into ourselves. Now, this process is expensive. It takes long, okay? How many horses can you go get and wait for them to make the insulin, isn't it? So by growing millions of bacteria inside one fermenter, you get so much insulin is much easier and costs way less, okay, than, you know, having to sedate the horse, extract it from the horse, things like that. Okay, let's go through step by step. Okay, number 1. Okay, this is how we do it. We take some human cells, okay, we liquidized it, okay. Then we add some chemicals in to make the DNA precipitate. Means it is just to make it easily separated out so that we can take out some of our DNA to put inside the bacteria. Now, I want you to memorize some terms. Okay, so let's zoom through here. You see, this is our DNA. You see that chain of double helix. Okay, so we will use an enzyme called restriction enzyme. Okay, this is the restriction enzyme. Okay, imagine it is like a scissors. It cuts off. See, once you put the restriction enzyme, then suddenly it becomes pieces here. Okay, so restriction enzyme is like scissors. It will cut off the DNA, and then you have sticky part at the end here. Okay, so now we take the sticky part. Okay, we will join, put together into the bacteria. Okay, we call this the plasmid. Okay, bacteria one, we don't call them DNA. We call that bacteria's DNA called plasmid. Okay, so we will add it in using another enzyme called ligase. So ligase is like the glue. Okay, so we glue in human DNA into bacteria plasmid. Remember? Okay, so restriction enzyme is to cut human DNA. Ligase enzyme is to stick human DNA into bacteria plasmid. Basically, bacteria plasmid is a circle of their own DNA. You just cut it out and put the human one. Okay, so actually the plasmid acts as a vector, putting the human insulin gene into the bacterium. So let's put it back to the bacteria like this. And then now this bacteria is genetically modified already, isn't it? And then bacteria, they produce asexually. It doesn't need to have any mate. So from one bacteria like this, it will produce the exact same copy of bacteria, genetically identical. And then from one bacteria like this, there's like millions of them. Okay, from that's genetically identical. We grow them in a fermenter. Then that's it. Once they're done growing, we take them out again. And then we harvest the insulin. Yeah, so that's how we get abundance of insulin. So I've already done with this whole chapter. There is this one part where I want you to read yourself about the issues of genetic engineering, what people think. Is it good? Is it bad? Yeah, that's more of thinking skills. Okay? Any question, please ask me. Yeah? All right, thank you guys.